The overall goal of this system is to study the reciprocal signaling, interaction, and responses between plant and microbe partners or plant responses to chemicals or abiotic factors in a simple experimental setup that closely mimics the natural environment. This video shows co-cultivation of plants with Agrobacterium tumefaciens bacterium.
This method can help answer key questions in plants' microbial interaction, microbial or plants' stress response, and molecular signaling, including complex signaling events at the initial stage of plants' microbial interactions. The main advantage of this technique is that it helps to understand the nature, signaling, and response of plants' microbial interactions in a simple setup that closely mimics the natural environment.
Though this method can be used to provide insight into plant interactions with a single microbial species, it can also be used to study plant interactions with multiple microbial species or with abiotic factors.
We had the idea for this method while searching for cell signaling homeostasis during plant microbe interactions that closely mimic natural situations.
To begin the experiment, seeds must be sterilized. For Arabidopsis thaliana, combine approximately 200 seeds with 500 microliters of deionized water in a microcentrifuge tube and vortex the tube at high speed. Centrifuge the tube at 9,000 times G for 30 seconds in a tabletop microcentrifuge. Using a pipette, remove the water supernatant. Next, add 300 microliters of 2% sodium hypochlorite.